biology classes. In last two videos of cell, the unit of life, we had discussed the structure and function of the plasma membrane as well as the cell wall, right? So today we will discuss the organelles that are present inside the cytoplasm. So there are various organelles and they have their distinct structure as well as functions. But there are certain organelles that are coordinated with each other with respect to their function. And this system of organelles is called as endomembrane system. So we'll see in detail about this system as well as organelles, their structure and function. So endomembrane system consists of endoplasmic reticulum, GOLGI apparatus, lysosomes and vacuoles. So these are the four organelles: endoplasmic reticulum, GOLGI apparatus or GOLGI complex, lysosomes and vacuoles. Okay, we see here endoplasmic reticulum is associated with the nucleus. GOLGI complex is associated with endoplasmic reticulum, and lysosomes and vacuoles are formed from the GOLGI complex or GOLGI apparatus. So here we can see this is the nucleus with the nuclear membrane associated with the endoplasmic reticulum. There are two kinds of endoplasmic reticulum: rough endoplasmic reticulum and smooth endoplasmic reticulum. GOLGI apparatus, which is associated with the endoplasmic reticulum, and from the GOLGI apparatus, lysosomes and vacuoles that will be formed. So that is the endomembrane system present throughout the cytoplasm in the eukaryotic cell. Endoplasmic reticulum was first reported by Porter and Thomson independently in 1945, and later on Porter named this organelle as endoplasmic reticulum. So if you see in this cell, we can see the continuous membranous system that is present throughout the cytoplasm, right? So it is extensive and metabolically active in certain cells, for example in pancreas or liver or the adipose cells, okay? But there are the organelles which are less in number in the spermatocyte, and endoplasmic reticulum is completely absent in some of the cells such as egg cells, then mature RBCs and the embryonic cells. Endoplasmic reticulum is the extensive network of membrane-lined channels-like structure. They form intercommunicating system of membranes. We can see here so many membrane, okay, lined channels that are present, and they run throughout the cytoplasm or protoplasm. So they are concentrated in the protoplasm and they will cover the large area of the cell. So they remain continuous with the plasma membrane, also with the nucleus, that is the nuclear envelope, and also with the organelle called as GOLGI complex or GOLGI apparatus, right? The endoplasmic reticulum have the distinct compartment called as luminal and extraluminal. Luminal is the one that is present inside the membrane-bound endoplasmic reticulum, and extraluminal is the one which is outside the endoplasmic reticulum. So we can see here the part which is present inside the endoplasmic reticulum is called as lumen. So therefore that compartment is called as luminal, which is present inside the endoplasmic reticulum. Whereas the part which is present outside the endoplasmic reticulum is called as extraluminal. So these channels or the membrane system of endoplasmic reticulum is filled with the fluid called as endoplasmic matrix. So that means this fluid is present inside the lumen or the cavity which the membrane has. So there are elements of endoplasmic reticulum: cisternae, vesicles and tubules. Cisternae are long, flat, sac-like structures. They lie parallel to each other in the cytoplasm. Vesicles are those which are round and oval-shaped membrane-bound sac-like structures, and tubules are those which are tube-like, irregular or branched structure. So all these three types of components or elements are present in endoplasmic reticulum. Okay. So there are two types of endoplasmic reticulum: rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum is usually called as granular, whereas smooth endoplasmic reticulum is called as agranular, that is without any granules. These granules are nothing but the ribosomes that are present on the membrane of endoplasmic reticulum. So the endoplasmic reticulum, which has ribosomes on them, on their membrane, is called as rough endoplasmic reticulum, whereas those endoplasmic reticulum which do not possess any ribosomes are called as smooth endoplasmic reticulum. So rough endoplasmic reticulum have the ribosomes. Okay, we should remember that ribosomes play a very important role in protein synthesis. Okay, so these ribosomes are present on the membrane of endoplasmic reticulum, performs the function of protein synthesis, and this rough endoplasmic reticulum is made up of cisternae, that is sac-like structures we can see, and also they have the tubules. Okay, it is also continuous with smooth endoplasmic reticulum and nuclear membrane. That means this endoplasmic reticulum is associated with the smooth endoplasmic reticulum and also the Golgi apparatus or Golgi complex. Okay, so we will see that endoplasmic reticulum is associated with the nucleus and nuclear membrane. Endoplasmic reticulum are of two kinds: rough endoplasmic reticulum and smooth endoplasmic reticulum. So rough endoplasmic reticulum is associated with smooth endoplasmic reticulum and also Golgi apparatus. Smooth endoplasmic reticulum, we can see in this structure, they are very smooth. That means they do not possess any ribosomes. So therefore they are agranular type of membrane. Okay. So these organelles, smooth endoplasmic reticulum, is formed of vesicles and tubules. You can see here they form sac-like structures called as vesicles, and also they will have tube-like structures. There are no cisternae as such which they have. Okay, they are continuous with the Golgi complex and plasma membrane. We can see here they are associated with the Golgi complex and also the plasma membrane. This is the plasma membrane. Here we should remember that the DNA is present in the nucleus, and the DNA is responsible to transcribe three types of RNA: rRNA, tRNA, mRNA. rRNA is ribosomal RNA, tRNA is transfer RNA, and mRNA is the messenger RNA. All these three types of RNA helps in synthesizing proteins, okay, which are very essential to carry out various metabolic activities. So the DNA which will transcribe these three types of RNA will be passed out from the nuclear membrane into the cytoplasm, because protein synthesis always occurs in the cytoplasm. So the ribosomes that are located on the endoplasmic reticulum will help in the protein synthesis. These proteins will be transferred inside the endoplasmic reticulum. There the folding as well as the modification of these proteins will take place, and then it will be released out of the endoplasmic reticulum. Okay. So rough endoplasmic reticulum helps in the protein synthesis and its secretion. Also it helps lysosome formation by the Golgi complex. Okay. Smooth endoplasmic reticulum, here the lipids will be synthesized. So smooth endoplasmic reticulum is meant for lipid synthesis. Okay. Rough endoplasmic reticulum is meant for protein synthesis. So we should remember that. It also plays important role in synthesizing ascorbic acid and also glycogen, for example in the liver cells, fats and steroid, for example in the adipose cells. So here it will be synthesized as well as it will help in the storing these materials. Okay. It also plays important role to release certain pigments, that is the visual pigments from vitamin A of retina of our eyes. Okay. In animal cells, the reproductive organs such as testis and ovary, here it helps in synthesizing some steroidal hormones such as estrogen, progesterone, testosterone, etc. So we came to know that endoplasmic reticulum will release proteins and smooth endoplasmic reticulum will release lipids. So the modification of the proteins as well as lipids will take place inside the endoplasmic reticulum and then it will be passed out of the endoplasmic reticulum. The next organelle is Golgi apparatus or Golgi complex that is associated with endoplasmic reticulum as well as the plasma membrane. Candelo Golgi named this organelle as Golgi apparatus in 1898. So it is the flat tube-like structures, network of tubules as well as vesicles and sac-like structures called as cistern. So this Golgi apparatus is also associated with the endoplasmic reticulum and the plasma membrane and it is located throughout the cytoplasm like endoplasmic reticulum. The Golgi bodies have four components: cisternae, tubules, vesicles and Golgian vacuole. These are the cisternae, that is the cis-cisternae, medial cisternae and trans-cisternae. We call these cis-cisternae as entry cisternae and trans-cisternae as exit cisternae. We see that these cisternae are like shallow bowls, right? This region is the convex part and this region is the concave part of the cisternae. Okay? So the entry phase is the forming phase, whereas the trans phase or exit phase is the maturing phase. Okay? So the Golgi apparatus have the forming phase and the maturing phase. So these are the cisternae, these are the tubules and these are the vesicles. So these vesicles are formed from the end part of these cisternae. Okay? And there are certain vesicles which will form the vacuoles called as Golgian vacuoles, which play important role to form lysosome, that is the third organ. This is the Golgi apparatus with the vesicles, right? So these vesicles will form the lysosomes, that is the third organelle. And some vesicles will secrete what is being taken by the Golgi apparatus. So we will recall here rough endoplasmic reticulum that forms the protein, which is modified and folding of protein is carried out by rough endoplasmic reticulum and then it will come out in the form of vesicles and it will get fused with the cis phase or forming phase of Golgi apparatus. Okay? And here in Golgi apparatus, various modification of these proteins will take place. On the other side, smooth endoplasmic reticulum where the lipids are synthesized, modified and then it is been transferred in the Golgi apparatus. So the vesicles which are formed from the endoplasmic reticulum will be fused in the cis phase or forming phase of Golgi apparatus. Okay. So in the Golgi apparatus, apparatus, the further modification of these proteins and lipids will take place, and they will release the vesicles from the trans phase or the maturing phase. These vesicles will secrete whatever is been modified from the Golgi apparatus. Okay? Whereas some of the vesicles will form the organelle called as lysosome. So Golgi bodies are made up of tubules. They are branched. The vesicles are formed at the end of the tubule, and they can be smooth or rough. And the Golgi vacuoles are formed from the trans phase or maturing phase of the Golgi apparatus, which will later form the third organelle called as lysosome. So Golgi bodies play important role in packaging the material which is been modified inside it. Okay, it will be sent in the cytoplasm, so therefore it is called as intracellular, and also it plays important role in secretion called as exocytosis, which will be released out of the cell. Okay, it also helps in the transformation of membrane, for example formation of vesicles which will later form lysosomes, and also the materials that are sent from the endoplasmic reticulum, they are being packed at the cis phase and then they are released at the maturing phase. So Golgi apparatus play very important role in synthesizing carbohydrates. Okay, so these carbohydrates will combine with the proteins to form glycoproteins, and the lipids that are being sent by the smooth endoplasmic reticulum will also bind with the carbohydrate and it will form glycolipids. So this process is called as glycosylation. Okay. Also, both lipids and proteins which have been sent to the Golgi apparatus, they both will also combine together to form lipoprotein, as the name says, lipids and proteins. Okay, so it forms lipoprotein and the process is called as liposylation. The next organelle is the lysosomes. Lysosomes are also called as digestive body. As the name says, digestive indicate that it helps to carry out the digestion process in the cell itself. Okay, and this was reported in 1955 by Christine D. Dewey. Okay, these lysosomes are formed by packaging process of Golgi apparatus we have already discussed, and they are membrane bound irregular vesicles having the enzyme called as hydrolyzing enzyme. These are the enzymes which play important role in digesting the material which comes inside these vesicles. Okay, and these enzymes are very active in the acidic condition or acidic pH that is less than pH 7. Okay, altogether there are more than 40 enzymes that are present in the lysosome. It depends what type of digestion it will carry out. Accordingly, those lysosomes will have the enzymes. Okay, lysosomes are of different kinds depending upon what structure they have and also what functions they will be performing. Primary, secondary, tertiary, also called as residual, and autophagic lysosomes. Primary lysosomes are those vesicles which are formed directly from the maturing phase, and they will have the enzyme, that is hydrolyzing enzyme. Secondary lysosomes are those which will be combining the lysosome, primary lysosome, and the phagosomes. The phagosomes are formed by the plasma membrane. Okay, whatever comes inside the cell, it may be water or any foreign bodies or any food material which enters inside. These plasma membrane will form vesicle-like structure and take that food material or water content inside the cell. Okay, so these vesicles filled with the foreign bodies or the water will combine with the lysosome. So these lysosomes will digest the foreign bodies that has entered inside. The third one is tertiary type. that is residual. As the name says residual, that means after the digestion of the food material that has entered inside the cell, the remains that will be present in the lysosome is the residual lysosome. Okay. So these remains that are present, that will be thrown out of the cell by the process called as exocytosis. So it will be secreted out of the cell. Okay. The fourth type is autophagic lysosome. Autophagic indicate it is autodigestion. That means these lysosomes will directly digest its own material that is present inside the cell. For example, any dead cells or the organelles which are of no use. So these organelles will be digested by the autophagic lysosomes. So usually it happens at the region where the injury has been taken place. So the dead cells or the other part of the cell which is not required by the cell will be directly digested by these lysosomes. So therefore it is also called as disposal units. After the digestion of these material inside these lysosomes, the product will be released in the cytoplasm so that they can form more organelles. Okay. So therefore these lysosomes are also called as recycling center of the cells. So the main functions are, it can digest the food content within the cell as well as outside the cell. That means it can be released out of the cell and there also digestion can be carried out. They play a very important role as the body defense or natural defense. One of the example is white blood cells or leukocytes, wherein the lysosomes will combine with the phagosome. As I have said, phagosome are the bodies which are formed by the plasma membrane, and these bodies will have any foreign body. So any foreign body, bacteria or virus which enters inside our body will be taken through the phagocytosis. That is, phagosome will be formed and that will combine with the lysosome and the digestion will take place. They are rich in all type of enzymes, say phosphatase, then lipase or the proteases or carbohydrates or nucleases. As the name says, lipases, they will break down the lipids, proteases will break down the proteins, carbohydrates will break down the carbohydrates, and nucleases will break down the nucleic acid. Okay, so they help in the metabolism of these material that is present inside this cell, and that is because of the enzymes that they have inside. We should also remember that these enzymes are active in the acidic pH. So the membrane which surrounds the lysosome will protect the other part of the cell, okay, from the acidic nature of the enzyme. Also we had seen that dead cells or debris that is present inside the cell, it will be destroyed and therefore it can be called as natural scavenging. Autolysis is one of the peculiar function of lysosome. They act as suicidal bags. Now as the name says, suicidal bags, that means they will destroy themselves as and when required. Okay. So here the lysosomes are usually surrounded by the membrane. So this membrane will get burst. So they help in destroying the cells or the tissues or organs that are not required by our body. The lysosomes will self-burst themselves and release the hydrolytic enzyme that is acidic in nature in the cell. So therefore, because of these enzymes, the cell will get destroyed. Okay. So therefore, it is called as the suicidal bag of the body. You can see these are the endoplasmic reticulum which will release the proteins and lipids in the Golgi apparatus. The vesicles that are formed from the endoplasmic reticulum will fuse with the cis phase of the Golgi apparatus where the modification will occur. So once the modification is been done, the lipoproteins or glycolipids, all glycoproteins will be released out, okay, from the Golgi apparatus. So up till now we have understood the functions of endoplasmic reticulum, Golgi apparatus and the vesicles. The fourth organelle is the vacuole. Vacuoles are formed after the fusion of many vesicles that are formed from the Golgi apparatus. So those are the vacuoles. Also we should remember that lysosomes are rare in plant cell and majority it is present in the animal cell. Plant cell, they have the cell wall for the protection which is absent in animal cell. So therefore lysosomes help in the protecting the cells. Vacuole, a large central vacuole is present in the plant cell, whereas in animal cell if it is present they will be small in size, right? So they occupy 90% of the plant cell. Indicate it plays very important role in the plant cell. So they are sac-like vesicles that are formed after the fusion of these vesicles. The vacuole have the membrane called as tonoplasm, which is a single membrane bound structure, okay? And it will store water, sap, excretory product and also other materials that are not useful for these cells, okay? So the tonoplast play very important role in transporting certain ions against the concentration gradient. When we speak about against the concentration gradient, it is from the region of lower concentration to higher concentration. So therefore it requires ATP energy. Here the tonoplast of the vacuole will take the ions that are not required by the cell and it will take forcefully inside there with the help of energy. So though the ions are less in the cytoplasm, still it will carry out the transport. So depending on the content that the vacuole have, okay, and the function it will be performing, they are of different kinds. The first one is sac vacuole. Sac is the fluid that is filled inside the vacuole. So it will have certain minerals, sugar, amino acid, proteins or the pigments such as anthocyanin which imparts the fruit color or flower color. Contractile vacuole are present in most of the protozoan members. One of the example is amoeba. If the water content is more inside the cell, so these contractile vacuole will get filled with that extra amount of water present inside the cell. And these water will be thrown out of the cell after fusion of these vacuole with that of plasma membrane. So this way the water will be released out of the cell. The third one is food vacuole. As the name says food, it will engulf the food material. So most of the protists as well as higher animals will see the ingestion of the foreign body or the food material. Okay, we also call it as endocytosis, which is brought inside the cell. Okay, so these food material or the foreign bodies are digested inside the body of these organisms. Okay, we had also seen here whatever foreign body that enters inside the cell, it fuses with the lysosome and lysosome have hydrolytic enzyme which will help in digesting these foreign bodies. Other than these, there are certain gas vacuoles which will help in the buoyancy, especially in the aquatic organisms. Also seen in the prokaryotic organism, they will store certain gases inside these vacuoles which will help in the buoyancy and mechanical support. So that was all about the endomembrane system. We have discussed regarding the four organelles, their structure and the function present in eukaryotic organisms or eukaryotic